Hey guys, it's Miss Roper. I told you I would make a webcast thing, so here it is. It's done in iMovie, and I'm going to try to upload it later. But we're going to go over chapter 20 in this one, and then I'll go over chapter 21 later because I don't know how long this is going to take me. So I will have this up as soon as possible, both of them. Okay, so we finished off on Wednesday talking about chapter 19, which was expression of eukaryotic genes and how they control the expression. So now we're going to start talking about how they have figured out how to control and express eukaryotic genes, and now what can they do with it to make it useful to study anything. So in chapter 20, we're going to go over DNA technology and how the study of genomics and how it's been useful in medical, in agriculture, in pharmaceutical, and in figuring out our big biology questions that we've always asked, like where do we come from, how different are we really at the genetic level, blah blah blah. So we're going to start talking about that. Now, there's a lot of technical stuff in this chapter, and it's really hard for me to talk about this without having like a slideshow. So I'm going to put up a slideshow too. But for right now, I'm just going to go over it in the book. Okay, so I'm going to just follow along with me in the book. I'm going to make sure that I tell you what chapter, what page number, what figure I'm on, okay? And I keep looking at this, and I should be looking at the little dot thing. So I'm not ignoring you. I'm trying to work this thing out. It's kind of weird. Okay, so in chapter 20, this is an old book. Okay, this is 2006. It's been 6 years. That's eternity for there to be new technological advances, and I can't even tell you, some of this stuff is just outdated. I mean, the computers that you see in the picture on the front page of chapter 20, yeah, those are like Jurassic Park old. So, a lot's changed since then, and we're just gonna have to deal with the fact that we just have just an old book. Okay, so there's not going to be a whole lot of questions on the AP exam over specifics of technology per se. They're probably going to ask you more like, oh, gel electrophoresis, expect you to know what that is, and then say, how has it helped us analyze something or other, something like that. Okay, so you really just need to know the applications behind everything, not so much the minute details. Okay, so let's start. All right. So it talks about some of the advances they've made in DNA technology. The most common one that you'll always hear about is genetic engineering. We've cloned, we've engineered, blah, blah, blah. All right, and this is the direct manipulation of all genes. Okay, so that means that you take them out, you knock them out, you put them in another organism, you recombine them, you study the sequence, you change around the sequence, something like that. Okay, it's any type of manipulation. Now, usually we manipulate them to mix them with other DNA from either another organism of the same species or an organism of a different species. Okay? Once we have two sources of genetic information, so DNA, then we call it recombinant DNA now because you're recombining two different types of DNA. Okay? So, and then the third big thing that they talk about is biotechnology. Basically it's telling you that it's mixing technology with biology. All right, so that's nothing new. But it's manipulation of organisms and their products to try and get something that is useful, like pharmaceutical-wise or environmentally or something like that. Okay, so the big one that we're going to talk about, though, is DNA cloning. Okay, and I don't just mean DNA cloning by Dolly. I don't mean making just whole organism clones. I mean, I'm talking about just making small inserts of genes that are cloned. Now, in the big scheme of things, when you're talking about products or when you're talking about proteins or enzyme function, you're usually talking about one gene. All right, and it's really hard when you have thousands and thousands of base pairs and all of these chromosomes. It's hard to isolate a specific gene. And so the scientists had to come up with a way to define where a gene starts and stops and be able to extract it so that they can use it for something. All right, and they found a way into this by looking at bacterial plasmids. Now you have to remember that plasmids in bacteria are smaller circular pieces of DNA that have a couple of genes on them that are unnecessary, not necessary for the function of the whole organism. Okay, so that would mean that it doesn't code for anything that, any enzymes that the organism has to have. It doesn't code for any proteins. It doesn't code for any other stuff that the organism can't live without. Okay, so it's extra stuff, and usually it's going to confer ampicillin resistance. Maybe it's got extra genes for more proteins, something like that. Okay, so usually it's going to be something that is not really necessary for the bacterial organism to survive, but actually helps it in harsher conditions. Okay. All right, so the first thing that they talk about, I'm looking at figure 20.1 on page 376. They're going to go through how they actually make, or the overview of how they clone DNA. Okay, so first of all, you have to know what gene you want. All right, so you pick out the gene of interest. Say it's the one for, I don't know, anything. Doesn't matter. Okay, so it's a specific gene that you want to replicate many, many times, but just that gene. So you pick out your gene of interest, and then you isolate your bacterial plasmid that you want to stick it into. Okay. Once you insert the gene of interest into the bacterial plasmid, you now have a recombinant DNA plasmid. Okay, because it's genes from two different sources. Okay. Then you take the plasmid, the recombinant DNA plasmid, and stick it back into the bacterial cell, and then it'll replicate, okay, as it grows, as it divides. And as it reproduces, it copies over and over again its own chromosomal DNA plus all the plasmids it has in its cell. All right, then the hard part about all this comes when you need to figure out which one replicated well and which one actually replicated the plasmid that has your recombination in it. All right, so not every single bacterial cell on the plate is going to have your recombination, so you have to go and screen for it. All right, and this is where the most tedious part is. All right, this is going to be the hardest part. Now, you would use bacterial plasmids to clone DNA for two reasons. Okay, number one, you want to look at some type of protein, okay, and how it functions, but you need to, rapidly make a whole lot of it. So you'd use bacteria. Second thing is that you want to produce many copies of a specific gene so that you can do some kind of analysis on it. Okay, whether it's running it through a gel or sequencing it to figure out the nucleotide base pairs or something like that. But you need many copies of it. Okay? So that's an overview. That's on figure 20.1. Now we're actually going to look at how you make recombinant DNA. Okay? Now, the first thing that you're going to have to do is make sure that you can isolate your specific piece. In the 1960s, they started to discover restriction enzymes, okay, in bacteria, of course. Okay, that's where they always discover everything. But restriction enzymes are enzymes that are specific to certain sequences on a DNA chromosome, and they will cut at those sequences, called a restriction site. Okay, it is limited in the number of specific sequences, and it will always cut at those specific sequences. Okay, so you could have a piece of DNA many, many times over. You introduce it to this restriction enzyme, it'll always cut at those sequences and those sequences only. Okay. Now, why do we need restriction enzymes, or why do bacteria need restriction enzymes? It's a defensive mechanism. It's a defensive mechanism. Okay, so they use them to make sure that they can recognize foreign DNA and cut it up so that it doesn't start to integrate into the genome. It's a short nucleotide sequence that they're going to be looking at. Okay, so they're going to be recognizing maybe 10 to 12 base pair long sequence, and they protect their own DNA by methylating it. Okay, so remember the methyl groups to silence genes? It's the same thing here. You stick a methyl group on the A's and the C's of a specific sequence, and it is physically impossible for the restriction enzyme to reach or read any of those sequences so that it cannot cut there. Okay, there's many, many types of restriction enzymes. There are a couple that are very generic that we use all the time. That'd be HinD3, EcoR1. There's some other ones. So we'll be looking at that more when we do our lab. So they recognize the restriction site, and they cut. Okay, and there may be many, many restriction sites, but they will always cut. And those little fragments that you get are called restriction fragments. All right, and those are the things, maybe it's a start and stop of a specific gene that you want. Okay,